This video will serve as an introduction to cell reproduction and the control or regulation of the cell cycle. When we think about the goal of cell reproduction, we have to think about a DNA molecule. Now, up until this point, we haven't talked a lot about DNA, so a pretty big part of this video will be an introduction to the structure of a DNA molecule. But suffice it to say that for now, in order for cells to divide, the DNA molecule must first be copied. And later on in the semester, we'll talk about the mechanism that's responsible for the copying of DNA. But for right now, just assume that a DNA molecule that might be present within a cell, whether that cell is prokaryotic or eukaryotic, is capable of being copied. So we're going from a cell with a single DNA molecule to a cell with two DNA molecules. And in the process of cell reproduction or cell division, a copy of that DNA molecule will be passed on to each of two new cells. We'll see later on that while many textbook authors, many website authors will tell you that these DNA molecules are identical to each other, that is rarely the case. So each of these two cells will have inherited a DNA molecule, but there is no guarantee that those DNA molecules will be identical to each other. So what is DNA? Well, DNA is a nucleic acid. It's the fourth category of biological molecule that we have seen thus far in AP biology. Remember that we've seen carbohydrates and lipids and proteins. We quickly addressed nucleic acids early in the first semester, but we didn't talk a lot about them. So this is going to be our opportunity to explore their structure and function in more detail. Nucleic acids are the source of genetic information within a cell. Whether you're talking about DNA or RNA, both of these molecules serve as genetic information. Now their function within the cell will be different, but both are serving as sources of information. RNA is often a single-stranded molecule, DNA a double-stranded molecule. Regardless, RNA and DNA are both polymers. And these polymers consist of monomers linked together to form long chains. The monomers that are used to build nucleic acid polymers like RNA or DNA are nucleotides. And every nucleotide consists of three parts: a sugar, ribose in RNA, or deoxyribose in DNA, a phosphate group, and then a combination of nitrogenous bases. In DNA, we're going to find adenine, guanine, cytosine, and thymine as nitrogenous bases. In RNA, uracil will replace the nitrogenous base thymine, but adenine, guanine, and cytosine will still be found in RNA. So if we begin to think about the structure of a nucleic acid, whether it's DNA or RNA, we have our nitrogenous bases, and we can see those nitrogenous bases come in two general categories: pyrimidines and purines. And what I'm hoping that you can see from right off the bat is that the pyrimidines are all single ring structures, while the purines are double ring structures. You will never have to memorize the structure of adenine or guanine or thymine, but it is helpful to know which nitrogenous bases belong to the category of pyrimidines and which nitrogenous bases belong to the categories of purines. I mentioned on the previous slide that the sugar component of the nucleotide is deoxyribose in DNA and ribose in RNA. And here we can see the difference between deoxyribose and ribose. In deoxyribose, a hydrogen atom has replaced the hydroxyl group at this position. And on the next slide, we'll talk a little bit more about these numbers that we can see along the structure of the molecule. But suffice it to say that deoxyribose is different from ribose in respect to the sugar, where that in deoxyribose, a hydrogen atom has replaced a hydroxyl group in the structure of the sugar molecule. So here we can see a more general depiction of the nucleotide. Remember, a nucleotide consists of a phosphate group, a sugar molecule, deoxyribose or ribose, depending on whether we're looking at DNA or RNA, and then a nitrogenous base. We can see that we've taken these nucleotides and we've linked them into a long polymer chain. This is not nearly as long as an actual DNA or RNA molecule, but I want you to focus in on the fact that here we have our phosphate group, here we have our sugar, here we have our nitrogenous base. We've linked those nucleotides together, and we've formed a polymer chain. I also want to call to your attention that at the end, or the ends of the polymer chain, we have very specific functional groups that will be present. At one end of the molecule, whether it's RNA or DNA, we're always going to have a phosphate group. And we're going to refer to this end of the molecule as the 5' end of the molecule because this is where the phosphate group will always be located. At the other end of the molecule, while it's not shown in this picture, but is shown in this picture, we're at the 3' end of the molecule. At the 3' end, we're always going to find a hydroxyl group. So on one end of the molecule, we'll find a 5' phosphate. At the other end of the molecule, we'll find a 3' hydroxyl. So this is a more general depiction of what we saw in the previous slide. This picture is showing us a molecule of DNA. Here we can see the traditional double helical structure of the DNA molecule, and if we zoom in on each part of the DNA molecule, we can see a picture very similar to the one that we saw in the previous slide. We can see the backbone of the molecule, one of the two strands that give rise to the double helical structure of DNA. We can see those phosphate, sugar groups that alternate all the way down the length of the backbone as those nucleotides were linked to one another, and we can see the nitrogenous bases. I like this picture because it shows the difference between the double-ringed purines, A and G, and the single-ringed pyrimidines, C and T. Down below, we can see what actually is happening along the other strand of the DNA molecule. Remember, DNA is a double-stranded molecule, while RNA is a single-stranded molecule. Here we can see that the nitrogenous base thymine is forming an interaction with the nitrogenous base adenine, while the nitrogenous base guanine is interacting with the nitrogenous base cytosine. This is going to be an important rule for us as we continue to explore the structure of DNA, that A always pairs with T and C always pairs with G. Well, let's take a look at the structure of the nucleotide in a little bit more detail. Previously, you saw the numbers next to the structure of the sugar in the picture. We can go back now and we can elaborate upon what those numbers actually represent. The sugars in a nucleotide, whether they're ribose or deoxyribose, consist of 5 carbon atoms. And we're going to number those carbon atoms with the numbers that you see here, 1, 2, 3, 4, 5. We're also going to give each of those carbon atoms a special designation. We're going to call them 1' or 2', 3', 4', or 5', simply to distinguish them from the carbon atoms that are going to be present in the nitrogenous base. So as we begin to make connections, I'm hoping you're looking at this molecule and you're saying to yourself, oh, I recognize this. This is a molecule of deoxyribose. Thus, it's a deoxyribonucleotide that might be found in a molecule of DNA. And I know that because of the 2' position, that 2' carbon atom, I have a hydrogen atom present rather than a hydroxyl group. If there was a hydroxyl group present in this position, I'd be looking at a ribonucleotide, a nucleotide that would be part of an RNA polymer and not a DNA polymer. So we've seen now that we can link nucleotides together to form longer nucleic acid polymers. At one end of our polymer chain, we're going to find a phosphate group attached to the 5' carbon atom. At the other end of our polymer chain, we're going to find a 3' hydroxyl group attached to the end of the polymer chain. So we have an orientation of that chain, 5' to 3' along its length. What we haven't talked about yet is the type of chemical bond that actually links the nucleotides together. We've seen the alternating phosphate-sugar molecules along the backbone of a strand of nucleic acid, whether it's DNA or RNA, but we haven't commented yet on the bond. And here you can see the type of bond that links nucleotides together. It's referred to as a phosphodiester bond because 2 ester groups, here's one and here's the other, are being used along with a phosphorus atom to form the chemical bond between the nucleotides. We'll see later on that in order to form this phosphodiester bond, we have to have a free 3' hydroxyl group present in order to attach new nucleotides to an existing nucleotide. And here's where we can see it. So what we're trying to do in this picture is link 2 nucleotides together with a phosphodiester bond. Here's the first nucleotide, and here's the second nucleotide. And what we're trying to do is attach the second nucleotide to the first nucleotide at the 3' hydroxyl group of the first nucleotide. So you can see that as long as we have this free 3' hydroxyl group, we'll be able to attach the second nucleotide to the first nucleotide, form that phosphodiester bond, and extend the length of the nucleotide chain, or the polymer chain, by one nucleotide. I did want to quickly highlight some of the other differences between RNA and DNA. We'll be talking more about RNA later on in the semester. I have already mentioned that uracil is going to replace thymine as the nitrogenous base in RNA. I've also mentioned that RNA is a single-stranded molecule and that the sugar ribose replaces deoxyribose found in DNA. And finally, I've also commented on the fact that at the 2' position, here it is again, we're going to find a hydrogen atom in deoxyribose, while we'll find a hydroxyl group at the 2' position of ribose. So these are some differences that distinguish DNA from RNA in respect to the two different types of nucleic acid. I've also shown you that A, the nitrogenous base adenine, tends to pair with the nitrogenous base thymine, and the nitrogenous base guanine pairs with the nitrogenous base cytosine. But what's responsible for holding this interaction together? Well, again, our good friend the hydrogen bond is going to be responsible for connecting these two nitrogenous bases together in either a molecule of RNA or a molecule of DNA. I wanted to point out briefly that 2 hydrogen bonds are going to hold together a pair of A and T nitrogenous bases, while 3 hydrogen bonds are going to hold together a cytosine-guanine base pair. So in general, cytosine and guanine base pairs are going to be considered stronger than an adenine-thymine base pair because 3 hydrogen bonds are stronger than 2 hydrogen bonds. But in general, what we're always going to be able to say is that adenine pairs with thymine, or a purine pairs with a pyrimidine, just like guanine, a purine, pairs with cytosine, the pyrimidine. Just one more quick look at the structure of a DNA molecule, again highlighting the hydrogen bonds, 2 hydrogen bonds between A and T base pairs, and 3 hydrogen bonds between GC base pairs. Well, that's going to serve as our introduction to the structure of DNA. We'll be building DNA molecules in class that I'm hoping will help solidify the structure of DNA in your mind. But ultimately, what we want to talk about is the role of DNA in cell division. So you know at this point that eukaryotic cells have nuclei, and hopefully you remember that DNA is found within the nucleus of eukaryotic cells. If we were in a prokaryotic cell, that DNA would simply be floating in the cytosol of the cell. You may also be familiar with the structure of a chromosome. And what I want to highlight now briefly is that a chromosome is nothing more than a coiled molecule of DNA, or a packaged molecule of DNA. And we can see that here. Here's the double helical structure of DNA. We've taken that structure of DNA and we've coiled it into the structure of the chromosome that you might be familiar with. And it's that chromosome molecule that's often found within the nucleus of a cell, or at least as depicted in the nucleus of a cell. One more piece of information that I wanted to share is that if we were to look at a DNA molecule that again can be considered as part of a packaged chromosome molecule, there are going to be regions of DNA that will serve as the genetic instructions to make a protein. And these subsets of the DNA polymer that serve as the instructions to make a protein are going to be classified as genes. Well, how is it DNA actually going to appear within a cell, whether it's in a prokaryote or a eukaryote? Well, it's going to appear in the form of chromatin. You can think of chromatin as a jumbled mass of both DNA and protein that ultimately, if it were packaged, could give rise to the structure of a chromosome. Let's take a look at what that looks like in a little bit more detail. So cellular DNA, again, whether you're in a prokaryote or a eukaryote, is going to come in the form of a chromatin molecule, that tumbled mass of DNA and protein. And believe it or not, chromatin is actually more DNA than it is protein. It's about a 60-40 split. A typical DNA molecule found in your cells is composed of 140 million nucleotides. Now, that can vary depending on the size of the chromosome, but up until this point, the length of DNA polymers that we've considered has been much smaller, something on the order of 10 nucleotides, nowhere near 140 million. Well, each of those individual nucleotides takes up space, and if we were to take a typical DNA molecule found within your cell and stretch it out, we'd have a molecule that's close to 5 centimeters in length. That's pretty big. If we took all of the DNA typically found in a human cell, we would actually have something on the order of 9 meters of DNA. And all of that molecular material has to be packaged not only within the cell, but within the nucleus of that cell if we're talking about a eukaryote. So how do we get all that DNA to fit inside the nucleus? Well, you guessed it, we're going to have to coil it up. And we'll look in a moment at an animation that summarizes this process, but suffice to say that in order to coil the DNA, we're going to have to wrap it around protein molecules. So remember that chromatin is 60% protein and about 40% DNA. So if we had a way to wrap the DNA around a protein component, we could begin to package the DNA into a much more smaller, compact structure. That's what you're seeing over here on the right-hand side. Here's our double helix. We're wrapping it around a collection of protein molecules. These proteins are called histones. And if we then had a way to take these DNA histone protein complexes and further compact them, we'd be on our way to building the chromosome that we saw on the previous slide. So let's take a look at what this packaging or this collection of packaging events looks like within the cell.
We'll see the remarkable way our DNA is tightly packed up so that 6 feet of this long molecule fits into the microscopic nucleus of every cell. The process starts when DNA is wrapped around special protein molecules called histones. The combined loop of DNA and protein is called a nucleosome. The nucleosomes are packaged into a thread known as chromatin. This fiber is then looped and further packaged using other proteins which are not shown here. The end result is that the DNA is tightly packed into the familiar structures we can see through a microscope, chromosomes. Chromosomes are not always present. They form around the time cells divide when the 2 copies of the cell's DNA need to be separated. At other times, as we can see now after the cell has divided, our DNA is less highly organized. It is still wrapped up around the histones, but not coiled in the chromosomes.
So hopefully you could see in that animation the process by which DNA molecules in the form of chromatin are packaged into the form of chromosomes. And I really like how at the end of the animation the narrator says that DNA typically is not found in the form of chromosomes. Only chromosomes will be present, or chromosomes will only be present as the cell is preparing to divide, preparing to reproduce. Most of the time, DNA will be in the form of chromatin, this loose, less compact mass of DNA and protein within the cell. Okay, so at this point, let's explore the structure of chromosomes in a little bit more detail. So if we think about a typical chromosome, you may be thinking about the X shape that you've seen in textbooks or videos or movies before. And again, while that is a valid interpretation of the structure of a chromosome, it's important to note that DNA is not in the form of a chromosome most of the time. Instead, it's in the form of that loose, non-packaged chromatin molecule. So if we think about chromatin having been packaged into the form of a chromosome, we're really only going to be able to see these chromosomes as a cell is getting ready to divide or is in the process of dividing. If we think about this X shape in a little bit more detail, each half of this duplicated chromosome is going to be composed of a sister chromatid. So each of these sister chromatids are going to be one individual DNA molecule, and we'll explore that phenomenon just a little bit more on a future slide. So if we think about a duplicated chromosome that has this X shape, each half of the X shape is going to be classified as a sister chromatid. Some textbooks will say that the two sister chromatids are attached to each other by a centromere, but this is a little misleading because in fact each sister chromatid has its own centromere, and the centromeres of those two sister chromatids will serve as the points of attachment for those two sister chromatids in a duplicated chromosome. So here's your sister chromatid, and here is the location of the centromere, but again, I would encourage you to consider the centromere as having, or as being a structure that both chromatids possess, that both DNA molecules possess, and serves as a common point of attachment for both of those DNA molecules within the duplicated chromosome. So we've seen this picture before as we introduced the video, and we said that at the beginning of the video, in order for a cell to reproduce, that DNA molecule must first be copied, and that each of those copies of DNA has to be passed on to the next generation of cells in order for the cell to divide successfully. If we were in a bacterial cell, we can think of this as the process of cell division as occurring when a single DNA molecule, remember if we're in a prokaryote, this DNA molecule is circular in nature, is going to be copied. And then very simply, if the cell divided in such a way that each of the new cells inherited one of those circular DNA molecules, we will have successfully divided the original parent cell into two new cells. Well, this works fine in prokaryotes, but what does this process look like in eukaryotes? Well, remember that in order for a eukaryotic cell to divide, the chromatin must first be packaged into a chromosome. And in reality, before the chromatin can be packaged, the DNA must be copied as well. So in this picture, the DNA has been copied and has been packaged into the form of this common duplicated chromosome. Remember, each half of the duplicated chromosome is a sister chromatid, and each sister chromatid has a centromere that serves as the point of attachment for the other sister chromatid in the duplicated chromosome. Now, if we had a way to separate these two sister chromatids from each other and thus pass one of these chromosomes into each of the two new cells produced through cell division, we would have successfully passed on a copy of the DNA to each of two new cells. So, in summary, if we started with a DNA molecule, and we copied it into the form of a duplicated chromosome, and then separated those sister chromatids so that each new cell received one of those sister chromatids, one of those DNA molecules, we would have successfully passed on the original copy of the DNA molecule to each of two new cells. This is just the beginning of our look at the process of cell division. So I'm hoping that this look at the structure of DNA and how the structure of DNA relates ultimately to the transmission of DNA molecules from one generation of the cells of cells to another has been helpful. We'll spend the rest of this week and the beginning of next week looking at what actually happens during a cell cycle. In other words, when a cell prepares for cell division, when it makes a copy of its DNA, when it finishes its preparation for cell division, and then actually commits to the process of division where each new cell inherits a copy of the DNA from the original parent cell.